You know, when I think about everything our brain does beyond just, well, seeing or moving, how it conjures up memories, dreams, even consciousness, it truly feels like magic sometimes.
It really does.
Today, we're going to pull back that curtain and really explore those higher level functions of the nervous system. We want to go as deep as we possibly can, aiming for that mastery level understanding of this incredible organ.
Yeah, let's do it.
Our source material for this expedition, this deep dive, comes from excerpts from BERNE LEVY Physiology, which is, a really foundational text.
Absolutely.
Our mission today is to really unravel the intricate world of the cerebral cortex and its many roles, moving beyond simple reflexes to the complex orchestrations that truly define, well, what it means to be human. We're aiming to master this information like the elite professionals in the field.
That's precisely it. And what's truly fascinating here is how our brain doesn't just process individual sensations, like a light flashing or a sound,
Right.
but brings it all together somehow to create that rich, unified experience of being human. We're not just talking about individual neurons firing, we're talking about the grand symphony they compose.
Yeah, the whole orchestra.
Exactly. So this deep dive will thoroughly explore its physical architecture, the very foundation, and then how it generates electrical signals, those incredible mechanisms by which we learn and remember, and also how it constantly adapts and rewires itself throughout life. It's quite something.
Okay, let's unpack this then. And let's start with the very foundation, the cerebral cortex itself. I mean, just fitting all that incredible processing power into your head. The brain's surface is brilliantly folded, isn't it?
It really is, like a giant, super efficient origami sculpture.
Yeah. These folds, the ridges called gyri and the grooves called sulci, or the deeper fissures, they aren't just for looks, are they?
Not at all. They pack in an enormous amount of neural real estate. It gives the human brain a volume of about 600 cubic centimeters, but a surface area of around 2,500 square centimeters.
Wow, that's huge.
It is. And that massive surface area is what enables the complexity of human thought, basically.
Okay, so maybe we don't need to get lost in every single crease, but there are some key landmarks that divide the brain into its functional zones, right?
Definitely. We should touch on those.
So we see key divisions like the central sulcus. That creates a pretty clear boundary between the frontal and parietal lobes.
Correct. Front and back, essentially.
And both of those are then separated from the temporal lobe, down below, by the lateral fissure. And then on the medial surface, the inside face of the hemisphere, you'll find the parieto-occipital fissure, separating the occipital and parietal lobes.
That's right, separating the visual processing area at the back.
What's cool, though, is that some parts of the cortex are actually hidden from view, aren't they?
They are indeed tucked away.
Like, buried deep within that lateral fissure is another lobe called the insula.
Involved in things like emotion and bodily awareness.
Plus, there's a whole group of structures forming the limbic lobe on the medial aspect, with its largest part, the hippocampal formation,
Crucial for memory.
folded into the perihippocampal gyrus of the temporal lobe. So you can't even see it from the brain surface until dissection.
Exactly. It's protected deep inside.
So to really understand how all these folds work at a deeper level, you mentioned there are different types of cortex.
That's right. It's crucial to know there are 3 main types, each with a distinct layered structure. Now, the star of the show for humans, by far, is the neocortex.
Okay.
It makes up approximately 90% of your brain and is characterized by its 6 distinct layers. This is where most of the heavy lifting for higher thought happens.
90%. Wow. What are the others?
Then we have the archicortex, which has 3 layers. The hippocampal formation we just mentioned, absolutely vital for memory, is a prime example of this type.
Simpler structure, then.
Relatively, yes. 3 layers. And lastly, there's the paleocortex, which has 4 to 5 layers. It's typically found at the border between the archicortex and neocortex, and often involved in things like olfaction, your sense of smell.
Okay, so let's zoom in on that dominant type, the neocortex and its 6 layers.
Yep.
Because you said each one has characteristic cells and a specialized role. This seems key for a deep understanding.
Absolutely fundamental. All right, layer 1, right at the top, is the molecular layer. It actually has few neuronal cell bodies,
Okay.
but it's dense with axon terminal synapsing on the apical dendrites. Those are the upward-reaching branches of neurons below, so it's a key input and integration layer.
Got it. Layer 2.
Layer 2, the external granular layer, primarily contains small neurons called stellate cells and some small pyramidal cells. It contributes significantly to intercortical communication, connecting different areas.
The communication between areas. Layer 3.
Layer 3, the external pyramidal layer. Here you'll find mostly small pyramidal neurons. These are crucial for associations between cortical areas, sort of linking related concepts or information.
Dissociations, right? Layer 4.
Now, layer 4, the internal granular layer, is really important. It's dense with stellate cells and a thick matrix of axons. This is essentially the brain's main incoming messages hub.
The main input receiver.
Exactly. It receives most of the sensory input coming up from the thalamus.
Okay.
Layer
Cool.
V, the internal pyramidal layer. This one is dominated by large pyramidal neurons, and these are the main source of efferent projections, the outputs from the cortex to most subcortical regions.
So this is the main output hub,
Yeah.
sending commands down.
Precisely, to places like the spinal cord for movement, the brainstem, the striatum. It's the primary output pathway.
And finally, layer 6.
Layer 6, the multiform layer. It's a bit more mixed, containing a mix of pyramidal, fusiform, and other cell types. Interestingly, these neurons project heavily back to the thalamus, the same areas that send input to the cortex.
Like a feedback loop.
Exactly, a crucial reciprocal loop that helps modulate the flow of sensory information. So you see this incredible layered processing. Layer 4V gets the main input. Layer V sends the main output. Others handle associations and feedback. It's incredibly efficient.
Almost like a highly specialized circuit board.
That's a great analogy. And within these layers, as you noted, there are two primary neuronal populations, or cell types. Pyramidal cells are the most abundant.
The ones shaped like pyramids.
Yep, exactly. They account for about 75% of neocortical neurons. Think of them as the primary communicators and the main output neurons. They're distinctively shaped, with that long apical dendrite reaching up like an antenna towards the surface to receive signals,
Okay.
and several basal dendrites spreading out near the cell body. Their axons project down into the subcortical white matter, and they release the excitatory neurotransmitter, glutamate.
So they're excitatory, they turn things on.
Generally speaking, yes. The larger pyramidal cells in layer V are responsible for those really long-range efferent projections we talked about to the spinal cord, brainstem, striatum, thalamus, mediating voluntary motor control, and other outputs.
And the layer 6 ones project back to the thalamus, completing that loop.
Correct. That reciprocal connection is vital. Now, the remaining 25% are the non-pyramidal neurons. These include stellate cells, which are star-shaped, and various GABAergic interneurons.
GABAergic means they use GABA, which is inhibitory.
Exactly. These are generally inhibitory neurons releasing GABA. They typically have shorter axonal projections, usually staying within the local cortical area, acting like local brakes or fine-tuners. Stellate cells, especially common in layer 4, can actually be either excitatory, glutamatergic, or inhibitory, GABAergic, and mostly project locally, helping to shape the processing within that layer.
So you have these main output cells, the pyramidals, and then these local circuit neurons, the non-pyramidals, sort of modulating the activity.
That's a great way to put it, modulating and refining the signals.
Okay, so how does information flow into and out of this incredibly intricate layered structure? Where does it come from?
Well, most input to the cortex from other parts of the central nervous system is actually relayed through the thalamus. Think of the thalamus as a major relay station.
Right, the big hub in the middle.
Pretty much. Now, thalamic nuclei that provide specific, topographically mapped projections, meaning a very precise point-to-point connection, like from your fingertip to a specific spot in the sensory cortex.
Like a dedicated line.
Exactly. Those primarily end in layer 4, the main input layer we discussed, but they also send some connections to layers 3 and 6.
Okay.
Then there are other thalamic nuclei, especially those relaying more diffuse input, say, from the brainstem reticular formation, the part involved in arousal.
Waking you up.
Right. These project more broadly and non-specifically, terminating mainly in layers R and the X. These diffuse projections are absolutely critical for globally modulating cortical activity, influencing overall states like sleep versus waking or general alertness.
So specific info goes mainly to layer 4. General arousal info goes wider, I and 6. Makes sense. What about input from other parts of the cortex itself?
Ah, yes. Beyond these subcortical inputs, every cortical region also receives a tremendous amount of input from other cortical regions. There are these massive fiber bundles, like the corpus callosum.
Connecting the two hemispheres.
Exactly. And the anterior commissure, too. These connect widely separated regions, even corresponding areas in each hemisphere, with their axons terminating mainly in layers I and 6, similar to the diffuse thalamic inputs.
Okay.
But here's something that might surprise you. The largest source of synapses connections in any given cortical region actually comes from local connections.
From within the same area or right next door?
Precisely, either from within that region itself or from its immediate neighbors. This really highlights the intense internal processing and communication happening within local cortical circuits. It's not just getting input and sending output, there's a huge amount of local chatter.
Wow, okay. And the outputs, where do they originate?
The efferent axons, those output cables, originate from the pyramidal neurons we talked about. The smaller pyramidal cells, mainly in layers 2 and 3, primarily project to adjacent cortical areas directly, and also to corresponding areas in the contralateral hemisphere via the corpus callosum. This facilitates that interregional cortical communication.
So layers 2 and 3 talk mostly to other cortex. Layer V talks mostly down to subcortical structures.
That's a good general rule of thumb, yes. It reflects that layered processing strategy.
What's truly remarkable then is how this architecture,
Yeah.
it isn't uniform across the whole brain, is it? It varies regionally.
Exactly. And that variation strongly reflects the functional specialization of different cortical areas. Scientists use several methods to map these variations and subdivide the cortex. Well, there's cytoarchitectonics, which looks at variations in cell density and structure, how packed the cells are, what types are prominent. Then there's myeloarchitectonics, focusing on axon density and size, how heavily myelinated or insulated the connections are. And also chemoarchitectonics, which examines the expression of specific molecular markers and neurotransmitter systems.
Different ways of staining or analyzing the tissue to see patterns.
Precisely. And these different approaches consistently reveal distinct functional zones.
And the most famously used map, I guess, is Brodmann's. Corbini and Brodmann divided the cortex into 52 discrete areas.
That's the one, numbered in the order he studied them. You probably recognize some of these numbers.
Yeah, like areas 3, 1, and 2 for primary somatosensory cortex, touch, and body sensation.
Right. Area 4 is primary motor cortex, initiating movement. Area 6 covers premotor and supplementary motor cortex for planning and coordinating movements.
Areas 41 and 42 for primary auditory cortex.
Processing sounds.
And Area 17 for primary visual cortex.
Exactly, and then subsequent studies have largely confirmed that Brodmann's areas are indeed distinct in their architecture, their connections, and their functions, although...
Is it still debated? Are the boundaries really that sharp?
That's a great question. While Brodmann's maps might suggest these sharp, immutable boundaries, the reality is often more nuanced. The transitions between many cortical areas can be quite subtle, and modern imaging often reveals more of a gradient than a hard line. But some areas do have quite distinct characteristics. For instance, the primary motor area, Area 4, and premotor areas, parts of Area 6, are often called a granular cortex.
Granular, meaning without granules.
Essentially, yes. It refers to the fact that they lack a clear, distinct layer 4, that main granular input layer. Which makes sense, right? Motor areas are primarily about output, not sensory input.
Ah, okay. Less need for a big input layer.
Exactly. And the primary motor cortex, Area 4, is particularly distinguished by these very large pyramidal cells in layer V, known as BET cells. They're huge and crucial for sending direct motor commands down the spinal cord.
BET cells.
Yeah.
Got it. So that's a granular. What's the opposite?
The opposite is granular cortex. Primary sensory cortices like the somatosensory, areas 3, 1, 2, auditory 41, 42, and visual 17 areas have a very prominent and dense layer 4 dominated by those stellate cells. That's why they're called granular.
Because layer 4, the granular layer, is so obvious.
Precisely. And the primary visual cortex, area 17, is even famously called the striate cortex because there's a noticeable horizontal stripe of myelinated axons running through layer 4, the stripe of Gennari, that you can actually see with the naked eye in unstained tissue.
Wow. A visible stripe.
Yeah. But it's worth clarifying when we say granular or granular, it's referring to the grouping of cell types into these distinct layers, particularly layer 4. It doesn't mean the overall percentage of pyramidal versus non-pyramidal cells changes dramatically. That stays roughly similar across most cortical areas.
Okay, so it's about the layering pattern, not the overall cell mix.
Exactly.
Now, beyond the neocortex, you mentioned about 10% as archicortex and paleocortex. What about their structure?
Right. The archicortex, like the hippocampal formation, which is so key for memory, has that simpler three-layered structure. You have the molecular layer, then a pyramidal cell layer, and a polymorphic layer. These roughly resemble layers 1, V, and 6 of the neocortex.
It's kind of like a condensed version.
In a way, yes. And remember, it's folded deep into the temporal lobe, so you only see it when the brain is dissected, highlighting its protected and vital role. The white matter covering it is called the alveus, which bundles up the hippocampal input and output fibers. The main output axons then coalesce to form the fornix, a major pathway connecting the hippocampus to other limbic structures.
Okay, and the paleocortex.
That one has 4 to 5 layers sitting at the border between the archicortex and neocortex. It's often involved in processing olfactory information, your sense of smell.
Right, smell being a very old sense evolutionarily.
Exactly.
So what does this all mean then? We've got this intricate structure, these layers, these different types of cortex. How does it all translate into the functions we experience every single day? How does this complexity work?
That's the big question, isn't it? How does structure enable function? We can think of the brain lobes as different districts, each with primary responsibilities.
Okay, let's explore those districts. Starting with the frontal lobe, the big one at the front. What's its main job?
The frontal lobe is heavily involved in motor behavior and what we call executive functions. You've got the motor cortex, premotor cortex, cingulate motor areas, supplementary motor areas, and the frontal eye field, all located here.
So planning, initiating, executing voluntary movements, including eye movements.
Precisely. And critically for us humans, Broca's area is usually located here in the inferior frontal gyrus of the dominant hemisphere.
Dominant meaning language dominant, usually the left.
Exactly. For over 90% of people, right or left-handed, it's the left. Broca's area is essential for generating speech, for articulation.
Okay, and what about the very front part, the prefrontal cortex?
Ah, the anterior prefrontal cortex. That plays a major role in your personality, your decision-making, planning complex behaviors, working memory, and emotional behavior. It's often called the CEO of the brain.
The executive suite.
You got it. And understanding its importance becomes really clear when you look at the clinical implications of damage.
What happens if that area is damaged, like through disease or those old lobotomies?
Right. Bilateral lesions of the prefrontal cortex damage to both sides can lead to really striking and often devastating deficits. People can have severe difficulties with attention, planning complex tasks, and problem solving. They might become very impulsive, exhibit inappropriate social behavior.
A complete personality change almost.
It can be. Interestingly, aggressive behavior often significantly decreases. And while the pure sensation of pain might remain intact, the emotional component of pain, the suffering part, is often significantly reduced. It's like they feel it, but they don't care about it as much.
Wow, that really highlights its role in integrating emotion and cognition.
Absolutely.
Okay, moving back from the frontal lobe, we hit the parietal lobe. What's its main role?
The parietal lobe is crucial for processing, perceiving, and integrating sensory information, especially somatosensory, touch, temperature, pain, pressure, but also visual and auditory information. This integration happens largely in the parietal association cortex.
The association cortex meaning it brings different things together.
Exactly. It connects extensively with the frontal lobe, allowing that sensory information to guide voluntary motor activity. Think about reaching out to grasp an object precisely. You need sensory feedback integrated with motor commands.
Makes sense.
And it can also transfer sensory information, somatosensory, visual, auditory, to language centers like Wernicke's area, which is vital for understanding language.
Okay. And specific lesions here also tell a story.
Oh, definitely. Damage to the left parietal lobe can cause something called Gerstmann syndrome. It's a really unique cluster of symptoms.
Like what?
Inability to name one's own fingers or point to named fingers on others, that's finger agnosia. Loss of the ability to do calculations, acalculia. Difficulty with writing agraphia, and trouble distinguishing left from right.
That's very specific. All from one area.
It highlights how localized some functions can be. Now, contrast that with lesions to the right parietal lobe, often from a stroke. This can lead to hemispatial neglect syndrome.
Neglect, what does that mean?
It means the patient profoundly neglects their left visual field and the left side of space. They lose awareness of that entire, their own body parts on the left, objects, events happening there.
So they might only eat food on the right side of their plate.
Exactly. Or only shave the right side of their face, or draw only the right half of an object. And sometimes they even deny their difficulties, which is called anosognosia. They're unaware of their own deficit.
Incredible. And you also mentioned mirror neurons being found here.
Yes, in the inferior parietal cortex, as well as the inferior frontal cortex. These are fascinating cells. They respond both when an animal performs a specific action like grasping something, and also when the animal observes the same action being performed by someone else.
So they mirror the action in the observer's brain.
That's the idea. This has led to compelling speculation that they might be the neural basis for understanding others' intentions, for empathy, and maybe even for learning new skills by observation. Human EEG studies have localized activity consistent with mirror neuron behavior to these areas too.
Fascinating. Okay, moving to the back of the occipital lobe. This one seems more straightforward.
Relatively speaking, yes. Its major function is overwhelmingly visual processing and perception. Brodmann Area 17, the primary visual cortex, is here, along with secondary visual areas 18 and 19, which handle more complex visual processing.
And damage here causes blindness.
Yes, but the visual field losses are often very precise depending on exactly where the lesion is. It's almost like a missing piece in their visual jigsaw puzzle.
How so?
Well, lesions of the cuneus gyrus, which is the upper part, result in blindness in the lower contralateral visual field. So damage to the right cuneus affects the lower left visual field.
Okay, contralateral meaning opposite side.
Right, and lesions of the lingual gyrus, the lower part, cause blindness in the upper contralateral visual field.
Very specific mapping.
Extremely specific. The occipital lobe also has important connections out to the frontal eye fields, helping to direct your gaze, and projections down to the midbrain that help control things like convergent eye movements, focusing on near objects, pupillary constriction, and accommodation adjusting the lens for near vision.
All crucial for focusing and directing our sight.
Exactly.
All right, last lobe, the temporal lobe, situated on the sides. You said this was a real multitasker and important for memory.
Absolutely. It handles the processing and perception of sounds via the auditory cortex and also vestibular information, that's your sense of balance and spatial orientation.
Okay.
It's also crucial for higher order visual processing. For example, the infratemporal cortex down below is heavily involved in recognizing complex objects, including faces. Damage there can cause prosopagnosia, the inability to recognize familiar faces.
Can't recognize faces.
Wow. And it also contains a specific part of the visual pathway called Meyer's loop. So unilateral temporal lobe lesions just on one side can lead to a very specific type of visual field loss, losing the upper quadrant of vision in both eyes on the side opposite the lesion.
Another specific visual sign.
Yes. But perhaps the most famous role of the temporal lobe, especially the medial part deep inside, involves the limbic system.
The limbic system, emotion and memory central, right?
That's the core of it. It plays a critical and complex role in emotional behavior, learning, and memory. It helps regulate emotional behavior partly through its influence on the hypothalamus via something called the Papez circuit.
Papez circuit. Sounds important.
It was a foundational concept for understanding emotion and memory. It's this loop projecting from the cingulate gyrus, part of the limbic lobe, to the entorhinal cortex and hippocampus,
Okay.
then via the fornix, a fiber bundle, to the mammillary bodies in the hypothalamus. From there, the mammillothalamic tract connects to the anterior thalamic nuclei, which then project back up to the cingulate gyrus, completing the loop.
A big circuit involving cortex, hippocampus, hypothalamus, thalamus.
Exactly. Now, while the Papez circuit is foundational, modern neuroscience understands that emotional processing is much more distributed. The hippocampus, and especially the amygdala, another key temporal lobe structure, are extensively connected to the prefrontal cortex, the basal forebrain, the anterior cingulate cortex, adding layers of complexity to that basic loop.
Got it. So damage to the temporal lobes, especially bilaterally.
Bilateral temporal lobe lesions can cause a really striking condition called Kluver-Bucy syndrome.
Kluver-Bucy. What does that look like?
It's characterized by visual agnosia. They can see objects, but can't recognize what they are visually. They might have a tendency to examine all objects orally, putting things in their mouth, even dangerous ones.
Wow.
Also, abnormal attention to irrelevant stimuli, sometimes hypersexuality, dramatic changes in dietary habits, eating things they wouldn't normally eat, and generally a decrease in emotionality. They often appear unusually placid or fearless.
That sounds like a profound disruption. What part is key?
While it was initially linked to large temporal lobe lesions, more recent studies really highlight the crucial role of the amygdala within the temporal lobe.
The amygdala, often called the brain's fear center.
That's a common label, yes. It's critical for conditioning the association of fear with painful or threatening stimuli. And it can trigger emotional or avoidance responses through its connections to the medial frontal cortex and anterior cingulate gyrus.
So it links stimuli to emotional responses.
Very much so. Plus, the amygdala projects to the nucleus accumbens, a region in the basal ganglia often called a reward center. The nucleus accumbens signals pleasurable events, partly in response to dopamine input from the ventral tegmental area, or VTA. So the amygdala isn't just about fear, it's deeply involved in processing the emotional significance of stimuli, both negative and positive, and motivating behavior.
A key player in emotion and motivation.
Absolutely. And clinically, we see these lobe specializations reflected in things like epilepsy auras.
Auras. The feeling someone gets before a seizure.
Exactly. They can be like a brief window into the function of the area where the seizure originates. For example, a seizure starting in the motor cortex might cause specific movements on the opposite side of the body, maybe starting in the hand and spreading up the arm. The Jacksonian march we mentioned earlier following the motor map.
Okay.
A seizure in the somatosensory cortex could cause an aura of tingling or numbness. One in the visual cortex might produce flashes of light, scintillations, or colors.
Auditory cortex.
Humming or buzzing sounds. Vestibular cortex, a sensation of spinning or vertigo.
And the temporal lobe.
Seizures starting in the temporal lobe association areas can cause more complex experiences, illusions, hallucinations, feelings of déjà vu, or sometimes, if the olfactory cortex near the uncus is involved, a really distinct, often unpleasant smell aura that's known as an uncinate fit. These specific auras are incredibly helpful for neurologists trying to pinpoint where seizures are starting.
It really maps function onto location in a very direct way.
It does.
We've talked about the brain's physical structure, the lobes, the layers, how different areas do specific jobs. But how do these incredibly complex operations actually happen? How does the brain think and feel moment to moment? This next part for me is absolutely fascinating because it reveals how we can actually peek into the brain's active life.
Through its electrical signals.
Exactly, using the electroencephalogram, or EEG. So the EEG is essentially picking up the brain's collective hum, right? It's a recording of the summed electrical activity.
Specifically, the summed extracellular currents that result from synaptic potentials occurring in the pyramidal cells. These are a type of field potential measured by electrodes placed right on your skull.
So not individual neuron spikes, but the combined flow of current around many neurons.
Precisely. The tiny currents from a single cell are far too small to be detected non-invasively on the skull. EEG waves reflect the combined, synchronized activity of many thousands or millions of similarly oriented pyramidal neurons. Their alignment allows their small extracellular currents to summate into a measurable field potential at the surface.
And you said it reflects synaptic potentials more than action potentials. Why is that?
It's primarily because synaptic potentials, the inputs to the neuron, have a much longer duration than action potentials, the outputs. We're talking milliseconds versus microseconds. This longer duration allows the electrical events from many synapses firing asynchronously or semi-synchronously to overlap in time and summate effectively.
Like longer waves adding up more easily than quick spikes.
That's a good way to think about it. If action potentials are like quick flashes of lightning, synaptic potentials are more like the slower, broader rumblings of thunder that collectively create the ongoing hum the EEG picks up.
Okay. Now here's something I've always wondered. Does a positive wave on the EEG mean excitation and negative mean inhibition?
That's a common misconception. It's actually not that straightforward.
Right.
Whether an EEG wave appears positive or negative on the scalp doesn't directly tell us if the underlying brain cells are being excited or inhibited.
Why not?
Because the sign of the potential depends on where the synaptic activity is happening relative to the electrode and the overall orientation of the neurons. For instance, excitatory input near the top dendrites of pyramidal cells might cause a negative potential at the scalp surface, while excitatory input near the cell body might cause a positive potential.
So the location matters.
Exactly. Inhibition at the dendrites could cause a positive wave, while inhibition near the cell body could cause a negative wave. So you can't just look at the polarity. Instead, neuroscientists look at the patterns, the frequencies, and the rhythms of the EEG waves, which reveal overall brain states.
Okay, patterns and rhythms. What are the normal ones?
Well, normal EEG tracings show waves of various frequencies, and these depend on things like your state of wakefulness, your age, where the electrode is placed, and whether there are drugs or diseases affecting the brain.
Right.
In a normal awake adult who is relaxed with their eyes closed, the EEG, especially over the parietal and occipital lobes at the back, typically shows a dominant rhythm called the alpha rhythm. That's around 8 to 12 hertz or cycles per second.
Alpha waves. Okay.
Yeah.
What happens if you open your eyes?
If you open your eyes or become more alert, that nice synchronous alpha wave tends to disappear. It gets replaced by lower amplitude, less synchronized, higher frequency waves, typically in the beta rhythm range, which is about 13 to 30 hertz. That's characteristic of an active processing brain.
So more alert means faster, less synchronized waves.
Generally, yes. Then there are slower rhythms like delta waves, 0.52 hertz, and theta waves, 3 to 7 hertz. These are typically observed during sleep, especially deeper sleep.
Delta and theta for sleep.
Yeah.
And sometimes you hear about EEG spikes. Are those action potentials?
Not necessarily. While they're called spikes because of their sharp, brief shape on the tracing, they don't automatically imply they are recordings of individual action potentials, especially in scalp EEG. They often represent synchronized bursts of synaptic activity in a population of neurons, particularly in conditions like epilepsy.
Okay, so spike is more about the shape on the graph.
Right. Now, beyond these ongoing background rhythms, we can also look at cortical evoked potentials, or ERPs, event-related potentials.
Evoked potentials.
Yeah.
Triggered by something specific.
Exactly. These are specific EEG changes that are elicited by a particular sensory stimulus, like a flash of light, a click sound, or a touch. They're best recorded over the part of the skull that lies directly over the cortical area being activated.
So a visual stimulus triggers a potential over the occipital lobe.
Precisely. And a somatosensory stimulus, like a tap on the wrist, evokes a potential best recorded near the junction of the frontal and parietal bones over the somatosensory cortex. These potentials reflect the synchronized activity of large numbers of cortical neurons, and sometimes even subcortical structures too, as they process that specific input.
But are they easy to see? Is the brain's response that clear?
Actually, the raw evoked potentials are often quite small compared to the ongoing background EEG waves, so they can be hard to pick out in a single trial.
So how do you study them?
We use a really clever process called signal averaging. You repeat the stimulus many, many times, maybe 100 or even 1,000 times, and you record the EEG epoch just after each stimulus. Then you electronically average all those recordings together.
Averaging. How does that help?
Because the evoked potential occurs at a fixed time after the stimulus each time, it's time-locked. When you average the records, this consistent signal adds up and gets stronger. The random background EEG activity, however, which isn't time-locked to the stimulus, averages out towards 0.
The signal gets amplified and the noise gets canceled out.
Exactly. It makes the evoked potential stand out clearly, revealing the brain's precise response signature to that stimulus.
And that's useful clinically.
Hugely useful. We use evoked potentials to assess the function of sensory pathways. You can test vision, hearing, touch, even in infants who are too young for a standard sensory exam. For instance, the initial components of the auditory evoked potential actually reflect activity in the brainstem pathway, allowing us to assess brainstem function non-invasively, which is critical in diagnosing certain neurological conditions or assessing brain injury.
So EEG isn't just about overall states, but also specific pathway function.
Right. And while it reveals normal brain states and pathways, it also serves as a powerful diagnostic tool for clinical abnormalities. This is where it really becomes crucial in neurology.
Okay, let's talk about that. How does EEG look in pathological circumstances?
Well, it becomes quite distinct and often diagnostic. During a coma, for instance, the EEG is typically dominated by very slow delta activity, indicating severely reduced cortical function and consciousness.
And brain death.
Brain death, which is the complete and irreversible cessation of all brain function, including the brainstem, is defined electrographically by a maintained flat EEG, essentially, no detectable electrical activity whatsoever, even with high amplification.
Okay. And epilepsy, you mentioned EEG is key there.
Absolutely central to diagnosis and classification. Epilepsy is characterized by recurrent seizures, which are basically episodes of abnormal, excessive, synchronized neuronal activity. The EEG shows specific abnormalities during and often between seizures.
You mentioned partial and generalized seizures earlier.
Right. In partial seizures, the abnormal activity starts in a localized area. If consciousness isn't lost, it's a simple partial seizure. We talked about the Jacksonian march, that stereotypical progression of motor activity reflecting the seizure spreading across the motor mat.
Like twitching, moving up the arm.
Exactly. If consciousness is impaired, it's a complex partial seizure, sometimes called psychomotor epilepsy. These often originate in the limbic structures of the temporal lobe and can cause illusions, hallucinations, or semi-purposeful automatic behaviors like lip smacking or fumbling with clothes. Scalp EEG recordings during these focal seizures, and often even between seizures, may show distinctive sharp waves or spikes localized to the affected area.
So the EEG can point to the seizure focus.
It can, yes. Now, generalized seizures involve wide areas of the brain right from the start and always involve a loss of consciousness.
Like petit mal and grand mal.
Correct. Petit mal, or absent seizures, involve a brief loss of consciousness, typically less than 15 seconds. The person might just stare blankly. The EEG during this is very characteristic and diagnostic. It shows generalized 3 Hertz spike in wave activity.
3 per second spike in wave. Very specific.
Very specific. Grand mal, or tonic-clonic seizures, are more dramatic. There's a longer loss of consciousness. The person often falls. It starts with a tonic phase increased muscle tone, stiffening followed by a clonic phase, rhythmic, jerky, convulsive movements. There might also be bowel or bladder evacuation. The EEG shows widely distributed high-amplitude seizure activity during these events.
A storm of electrical activity across the brain.
Pretty much. And even between these major seizures, the EEG in people with epilepsy often shows what are called interictal spikes, those sharp discharges occurring between seizures, indicating an irritable focus in the brain.
What's happening at the cellular level during these spikes or seizures? What causes that runaway activity?
That's a huge area of research. Experimentally, these interictal spikes seem to correlate with something called a paroxysmal depolarization shift, or PDS, in individual cortical neurons.
Depolarization shift.
It's an abrupt, unusually large, and long-lasting depolarization of the neuron's membrane potential, which triggers a burst of repetitive action potentials riding on top of it.
Like the neuron gets stuck in an on state.
In a way, yes, a hyper-excitable state. Several cellular mechanisms are thought to contribute to this in epileptic foci. One is the potential for regenerative calcium-mediated action potentials occurring in the dendrites of cortical neurons, adding extra excitatory drive. Another major factor is often a reduction in inhibitory interactions within the cortical circuits. The GABAergic brakes aren't working properly.
So not enough inhibition to balance the excitation.
Exactly. Plus, other factors can contribute, like electrical field effects from many neurons firing together, and the release of potassium ions and excitatory amino acids, like glutamate, into the extracellular space by hyperactive neurons, which can then further excite neighboring neurons, creating a vicious cycle. Understanding these intricate cellular mechanisms is absolutely crucial for developing more effective anti-epileptic drugs.
It really highlights the delicate balance between excitation and inhibition needed for normal brain function.
Absolutely. It's a constant dynamic balance.
It's incredible how EEG lets us diagnose conditions like epilepsy by seeing that balance go awry. But the EEG also reveals the brain's most profound, regular, and maybe still mysterious state. Sleep.
Ah, yes, sleep.
We spend a third of our lives doing it. But what is sleep, really?
Yeah.
And what happens in the brain when we transition into this altered state?
Well, we know our bodies operate on a circadian periodicity and internal biological clock. It naturally runs on a cycle that's actually a bit longer than 24 hours, maybe around 25 hours.
25 hours, but the day is 24 hours.
Exactly. So it normally gets entrained or synchronized to the 24-hour day-night cycle by environmental cues, primarily the light-dark cycle.
Light resets the clock each day.
Essentially, yes. And we can see characteristic changes in our EEG that correlate beautifully with changes in our behavioral state across this cycle. As we discussed, when you're awake and aroused, beta wave activity dominates that desynchronized low-voltage, high-frequency pattern.
The busy brain signal.
Right. But when you relax, close your eyes, your EEG transitions to that more synchronous, slightly slower alpha wave pattern, especially at the back of the head.
Okay, so that's the transition towards sleep. What happens next?
As a person actually falls asleep, they sequentially pass through 4 stages of what we call slow wave sleep. Stages 1 through 4 over about 30 to 45 minutes. Think of it as a gradual deepening of rest, each marked by characteristic EEG changes.
Stage 1.
In stage 1, the alpha waves start to become interspersed with lower frequency theta waves. You're drowsy, easily woken.
Stage 2.
Stage 2 sees further slowing of the waves, but the slow wave activity is interrupted by 2 distinctive features. Sleep spindles, bursts of activity around 12 to 14 hertz, thought to be involved in memory consolidation.
Consolidating memories while you sleep.
That's one leading hypothesis. And the other feature is K-complexes, large, slow potentials that might help suppress external stimuli and maintain sleep.
Okay, spindles and K-complexes in stage 2. What's stage 3?
Stage 3 sleep is primarily defined by the emergence of larger amplitude, much slower delta waves. You still see occasional sleep spindles, but the delta waves start to dominate.
Getting deeper. And stage 4.
Stage 4 is the deepest stage of slow-wave sleep. The EEG is almost completely dominated by high-amplitude delta waves. There are no sleep spindles here. It's very hard to wake someone from stage 4 sleep.
So progressively slower and more synchronized brainwaves as sleep deepens. What else changes in the body during slow-wave sleep?
Your muscles relax significantly, though you still make intermittent small posture adjustments to avoid discomfort. Your heart rate and blood pressure decrease. And interestingly, your gastrointestinal motility, the movement of your gut, actually increases.
Huh. Rest and digest, literally.
Pretty much. And as mentioned, it becomes progressively harder to awaken someone as they move through these stages. When you awaken naturally, you typically pass back through the stages in reverse order, sort of preparing the brain for wakefulness.
But that's not the only kind of sleep, is it? There's also REM sleep.
Exactly. Roughly every 90 minutes or so throughout the night, slow-wave sleep transitions into a remarkably different state. Rapid eye movement, or REM sleep.
What's different about it?
Well, paradoxically, during REM sleep, the EEG becomes desynchronized again. It shows low voltage, fast activity, much like the pattern you see in an awake, alert individual.
So the brain looks awake, but you're asleep.
Precisely. That's why it's often called paradoxical sleep. Your brain is highly active, but your body is profoundly still.
To still how?
One of the key features of REM sleep is a complete loss of muscle tone, or atonia, in most of the body's skeletal muscles. You're essentially paralyzed except for phasic muscle contractions, most notably in the muscles that move the eyes, hence the name rapid eye movements.
The eyes are darting around under the eyelids.
Exactly. Many autonomic changes also occur. You lose normal temperature regulation, your pupils constrict, meiosis, and in males, penile erection typically occurs. Heart rate, blood pressure, and respiration also become much more variable and irregular compared to slow-wave sleep.
And this is when we dream most vividly.
That's right. While it's difficult to arouse someone physically from REM sleep, internal arousal is high, and this is the stage where most complex, narrative, vivid dreaming occurs. If you wake someone during REM, they're very likely to report a dream.
Fascinating. So this whole cycle, slow-wave sleep, REM sleep, repeats through the night.
Yes, typically in cycles of about 90 minutes, with REM periods tending to get longer towards the morning.
You mentioned the circadian rhythm is set by a biological clock and trained by light. Where is this clock?
The source of this precise circadian rhythm appears to be a tiny region in the hypothalamus called the suprachiasmatic nucleus, or SCN. It's located just above the optic chiasm, where the optic nerves cross.
Right above the eye input pathway. Convenient.
Very convenient. The SCN receives direct projections from specialized cells in the retina that detect ambient light levels. The neurons within the SCN seem to form this endogenous biological clock that adapts to the light-dark cycle, effectively setting your internal timing for a whole host of bodily functions.
So the SCN is the master clock.
It seems to be, yes. You directly experience the disruption of its normal entrainment when you're isolated from time cues, or when you travel rapidly across time zones, that's jet lag. And experimentally, destruction of the SCN in animals disrupts numerous biological rhythms, including the sleep-wake cycle, hormone secretion patterns, and body temperature cycles.
Given how fundamental sleep is, do we actually know why we sleep? What its purpose is?
You know, it's quite surprising, but despite its obvious importance, we spend a third of our lives doing it, and extreme deprivation can be fatal. The precise mechanisms controlling sleep and its exact purpose are still incompletely understood. That remains one of the big mysteries in neuroscience.
Really? What were the old ideas?
Historically, sleep was often thought of as a passive process, maybe just caused by reduced activity in the reticular activating system, that brainstem region involved in arousal. Less activation meant you fell asleep.
Like turning down a dimmer switch.
Kind of. But current data strongly suggests sleep is a highly active and carefully regulated process, not just a passive shutdown. For example, experiments show that anesthesia applied to the lower brainstem can actually cause arousal, not sleep. And stimulating certain parts of the medulla, near the nucleus of the solitary tract, can actively induce sleep.
So active sleep-promoting centers.
It seems so. Researchers have explored complex brainstem networks involving neurotransmitters like serotonin, norepinephrine, and acetylcholine. Manipulating the levels of these neurotransmitters can profoundly affect the sleep-wake cycle, suggesting they play key roles, but a complete, detailed neurochemical explanation for sleep onset and maintenance is still being worked out.
And the purpose? Why go through all this?
That's the other big question. It must have profound value, given the time investment and the consequences of deprivation. One prominent hypothesis, which we touched on with sleep spindles, is that sleep, particularly certain stages, is crucial for memory consolidation.
Strengthening memories.
Yes, the idea is that during sleep, short-term memories initially encoded during wakefulness, perhaps involving patterns of activity in the hippocampus and related temporal lobe areas, are actively reactivated and replayed. This process might help transfer them, integrate them, and stabilize them in cortical areas for long-term storage.
So sleep helps you learn and remember.
That seems very likely. There's a lot of evidence supporting that connection. But other functions, like perhaps restoring metabolic balance, clearing out waste products from the brain, or regulating energy, are also being investigated. Clinically, of course, sleep disorders are a major issue, ranging from chronic insomnia and narcolepsy to sleep apnea, bedwetting, sleepwalking, all highlighting sleep's critical role, not just for rest, but for overall cognitive health, attention, focus, and learning.
A huge essential part of brain function we're still trying to fully understand.
Absolutely.
Okay, let's shift gears slightly. The brain is clearly a unified whole, processing all this information, but it's also wonderfully specialized, isn't it? Especially between the two hemispheres.
Indeed. The concept of cerebral lateralization, or dominance, is fascinating.
We talk about cerebral dominance, which usually refers to language, right?
Yeah.
Assigned to the hemisphere where the main language centers reside.
That's the standard definition, yes. And what's truly striking is that in over 90% of people, both right-handed and left-handed individuals, the left hemisphere is the dominant one for language.
So even if you're left-handed, chances are your language processing is mostly happening on the left side.
That's correct. Handedness and language dominance aren't perfectly correlated, though left-handers do have a slightly higher incidence of right hemisphere or bilateral language representation. It's still a minority. Left hemisphere dominance is the norm.
And the evidence for this left dominance is pretty strong.
Extremely strong, mostly from clinical observations. Lesions, damage from stroke, tumors, injury in the left hemisphere consistently produce language deficits known as aphasia.
Aphasia, difficulty with producing or understanding language.
Right. We also see this confirmed temporarily during a procedure called the WADA test. A short-acting anesthetic is injected into the left carotid artery, which supplies blood to the left hemisphere. In dominant individuals, this transiently causes aphasia. They can't speak or understand language for a few minutes.
Wow. And if you do the same on the right side?
If you inject the anesthetic into the right carotid artery in someone with left hemisphere dominance, it usually doesn't substantially affect their core language abilities. It might affect other things, like the emotional tone of their voice or spatial reasoning, but not basic language processing.
Okay, so which specific areas in the dominant left hemisphere are most critical for language? We mentioned Broca's earlier.
Yes, two main areas are traditionally highlighted. Wernicke's area is a large region located in the posterior part of the superior temporal gyrus, often extending back into the parietal lobe.
Wernicke's area. What happens if it's damaged?
Damage there typically results in receptive aphasia, also called Wernicke's aphasia or fluent aphasia. People with this have profound difficulty comprehending both spoken and written language. Their own speech production might remain fluent, grammatically okay even, but it's often meaningless or nonsensical, filled with errors, made-up words, sometimes called a word salad. They don't seem to realize their speech doesn't make sense.
Fluent but meaningless.
Okay.
And Broca's area.
Broca's area is located more anteriorly in the posterior part of the inferior frontal gyrus, actually quite close to the part of the motor cortex that controls the face, lips, and tongue muscles.
Near the motor output for speech. Makes sense.
Exactly. Damage to Broca's area causes expressive aphasia, also called Broca's aphasia or non-fluent aphasia. Individuals with this have great difficulty generating speech and writing. Their speech is often slow, effortful, halting, and lacks grammatical structure, like talking in short telegraphic phrases. However, their understanding of language generally remains relatively good, much better than their production.
Difficulty getting words out, but understanding is okay.
Relatively speaking, yes.
Now, you sometimes hear these called sensory and motor aphasia. Is that accurate?
Those terms are often used, but they can be a bit misleading. It's important to clarify.
How so?
Well, a person with receptive Wernicke's aphasia doesn't necessarily have an auditory or visual impairment. Their ears and eyes might work fine. Their difficulty lies specifically in processing the language itself, extracting meaning from the words and sentences they hear or read.
It's a language comprehension problem, not a basic sensory problem.
Precisely. Similarly, someone with expressive Broca's aphasia might have perfectly normal motor control of their vocal cords, tongue, and mouth muscles for non-speech actions, like chewing or swallowing. Their difficulty is specifically with planning and executing the complex motor sequences required for speech production or writing. Aphasia is fundamentally a language processing impairment, centrally located in the brain, not a peripheral sensory deficit or a simple muscle weakness.
Okay. What's the term for just a mechanical difficulty with the speech muscles?
That's called dysarthria. That's an articulation problem due to impaired muscle control, perhaps from a lesion in the motor cortex affecting the face representation or damage to cranial nerves. Someone with dysarthria might have slurred or unclear speech, but their language comprehension and ability to formulate thoughts and write could be perfectly intact. It's a crucial distinction for diagnosis and therapy.
Language processing, aphasia versus speech mechanics, dysarthria. Got it. So we have these specialized areas, mostly in the left hemisphere. How do the two powerful halves of the brain talk to each other? How is information shared?
Interhemispheric communication, sharing information between the two sides, mainly happens through the corpus callosum.
That massive bundle of fibers connecting them.
Exactly. It's huge, estimated to contain about 200 to 250 million myelinated axons connecting corresponding regions in the two hemispheres. But there are also other smaller connecting pathways called commissures. The anterior commissure connects parts of the temporal lobes and some limbic structures. The hippocampal commissure connects the two hippocampi. They play a role, but the corpus callosum is by far the major highway for information transfer between the cortices.
And we know the corpus callosum is important because...
Its importance is dramatically revealed through both animal experiments and studies of human clinical cases where it's been damaged or surgically cut.
Cut? Why would you cut the corpus callosum?
It's sometimes done as a last resort treatment for severe intractable epilepsy to prevent seizures starting on one side from spreading across to the other hemisphere and causing a major generalized seizure. These are the famous split brain patients.
Okay, so what do studies on animals or these split brain patients show?
They show incredibly clearly how information is shared or not shared when the callosum is intact versus when it's cut. For example, classic animal studies looked at visual discrimination learning.
Learning to tell shapes apart.
Right. If an animal with an intact optic chiasm, where some visual fibers cross, and an intact corpus callosum learns a task with one eye covered, the information gets to both hemispheres. If you then test them using the other eye, they can still perform the task perfectly. The learning transferred.
Makes sense.
Even if you cut the optic chiasm before training, preventing direct visual crossing, the information still transfers between hemispheres, presumably through the corpus callosum.
Okay.
But, and here's the key part, if you cut both the optic chiasm and the corpus callosum before training the animal with one eye covered, then the information does not transfer. The hemisphere connected to the trained eye learns the task, but the other hemisphere remains completely naive. Each hemisphere has to learn the task independently.
It literally creates two separate learning systems in one head.
Essentially, yes. And similar, really powerful insights come from studying human split brain patients. Because humans have language, we can test them in ways that reveal the specialization even more clearly.
How do you test them?
Typically, you control the visual input. By having the patient fixate on a central point, you can flash a picture or a word, very briefly, to just their left visual field or just their right visual field.
So, the info only goes to one hemisphere initially.
Exactly. Information from the right visual field goes to the left hemisphere, and information from the left visual field goes to the right hemisphere.
Okay, so what happens?
If you flash a picture, say of a key, to the patient's right visual field.
Goes to the left hemisphere.
The
language hemisphere.
Right. The patient can easily say, I saw a key, no problem. But if you flash the picture of the key to their left visual field.
Goes to the right hemisphere, the non-language one.
Correct. Now, if you ask the patient what they saw, they will typically say, I didn't see anything.
Nothing, but they saw it.
The right hemisphere saw it, but it can't speak. The information is stuck over there. It can't cross the severed corpus callosum to reach the language centers in the left hemisphere.
Wow. But does the right hemisphere know what it saw?
Yes. If you ask the patient to reach under a screen with their left hand, which is controlled by the right hemisphere, and pick out the object they saw from a group of objects, they can correctly pick out the key.
So the right brain saw the key, controlled the left hand to find it, but couldn't tell the left brain what it was in words.
Precisely. It's a stunning demonstration of hemispheric specialization and the role of the callosum. This extends to other things
And
too.
motor commands.
Yes. If you give a verbal command, like raise your right arm, the left language hemisphere understands and can send signals to the ipsilateral motor areas controlling the right arm. The patient raises their right arm. But if you say, raise your left arm,
The left hemisphere understands, but can't tell the right hemisphere's motor cortex to move the left arm.
exactly. Without the corpus callosum, the verbal command processed in the left hemisphere cannot influence the motor areas in the right hemisphere needed to move the left arm. The patient typically won't raise their left arm to a verbal command.
And sensory information. Touch.
Same principle. They can verbally describe an object placed in their right hand. Info goes to the left hemisphere. But they often cannot describe an object placed solely in their left hand. Info goes to the right hemisphere, even though they can likely identify it nonverbally with that hand. The sensory information from the left side is cut off from the language centers.
It's like having two separate conscious minds almost.
It certainly raises profound questions about consciousness and unity of self. And it's not just language where these specializations become clear in split-brain patients.
What else is the right hemisphere good at then?
Studies suggest the right hemisphere often excels at tasks involving spatial reasoning, like solving 3D puzzles or arranging blocks to match a pattern. It also seems more involved in processing emotional cues, like facial expressions and body language, and understanding the emotional tone or intonation of speech, prosody. It's almost like it has its own distinct set of nonverbal, perhaps more holistic or spatial intelligence.
So left for language, logic, analysis.
Mm.
Right? For spatial, emotional, holistic processing. Is that too simple?
It's a common simplification, and there's some truth to it, but the reality is always more complex and interconnected when the brain is intact. However, these split brain studies really highlight those tendencies. They also reveal what happens when that connection is broken, a profound lack of normal interhemispheric coordination.
Lack of coordination, like what?
You might literally see one hand performing an action, like buttoning a shirt, while the other hand, controlled by the other hemisphere, simultaneously tries to unbutton it. Or one hand reaches for a desired object while the other hand pulls it away.
Whoa. Conflicting intentions from the two sides.
Exactly. It vividly demonstrates how the two hemispheres can operate quite independently when their primary communication link is severed, with one hemisphere expressing itself verbally and the other non-verbally through actions. It really puts a new spin on that feeling of being of two minds, doesn't it?
It really does.
And this helps us deeply understand conditions like the hemispatial neglect syndrome we mentioned earlier, resulting from damage typically to the right parietal cortex. The patient ignores the left side of space, might deny their own left limbs. It powerfully highlights how cutting off that crucial communication or damaging those integration centers leads to profound sensory and motor deficits as the brain struggles to construct a coherent, unified view of the world and the self within it. Anosognosia, the lack of awareness of the deficit, might even be related to this disconnection, the language hemisphere literally not getting the message about the problem.
Okay, that connection piece is huge. Now, this is where the rubber really meets the road for the learner in all of us, right?
Mm.
How do we actually acquire knowledge, store it as memory, and how does our brain literally change with experience? This feels central to mastering how the brain works.
Absolutely. This gets to the heart of adaptation. We define learning, neurobiologically, as the neural mechanism or process by which an organism's behavior changes as a result of experience.
The change mechanism.
Right. And memory is the storage mechanism for what is learned, the way that acquired knowledge is retained and can be accessed later.
Storage and retrieval. How do scientists even begin to study something so complex in our brains?
It's incredibly challenging, given the sheer number of neurons and synapses, trillions of connections in the human brain. So researchers often use several approaches. One is animal studies, sometimes using much simpler nervous systems.
Like slugs or flies?
Exactly. The marine mollusk aplysia, a type of sea slug, has been particularly informative because it has a relatively small number of very large, identifiable neurons, allowing researchers to map out specific circuits involved in simple forms of learning.
What other approaches?
Well, lesion analysis in both humans, like patients with brain damage, and animals is crucial, seeing what functions are lost when a particular area is damaged. And increasingly sophisticated cellular and pathway-level studies using techniques like electrophysiology, imaging, and molecular biology to look directly at synaptic changes.
Okay, tell me more about those aplysia studies. What basic learning did they reveal?
They've been incredibly insightful, providing fundamental building blocks. They clearly demonstrate three basic types of non-associative and associative learning at the synaptic level. First, there's habituation.
I'm getting used to something.
Exactly. It's learning not to respond to repetitions of an insignificant stimulus, like you tune out the constant hum of the refrigerator eventually. In aplesia, this involves a gradual decrease in the amount of neurotransmitter released from the sensory neuron onto the motor neuron with repeated stimulation.
Less signal gets sent.
Right. This seems to be largely due to the inactivation of presynaptic calcium channels, which are needed for transmitter release. And if the habituation is long-term, there can even be structural changes, like a reduction in the actual number of synaptic endings or active zones where transmitter is released.
Wow, physical pruning of connections.
It appears so. Second, we see sensitization. This is an increased responsiveness to innocuous stimuli after you've experienced a strong or noxious, harmful one.
Like being jumpy after hearing a loud bang.
Precisely. A strong stimulus can enhance the synaptic connections in related pathways, making them more responsive for a period. And third, aplesia exhibit associative conditioning learning to associate two stimuli.
Like Pavlov's dogs associating the bell with food.
Exactly that principle. Learning to respond to a previously insignificant event, like a light touch, after it has been repeatedly paired with a significant one, like a shock. These basic mechanisms, habituation, sensitization, association discovered in aplesia, are believed to represent fundamental processes conserved across many species, including us.
Simple systems revealing basic rules. Okay, moving up to vertebrates, like us. What are the key mechanisms there?
A major focus of research in vertebrate learning and memory has been on dynamic changes in synaptic strength, the idea that connections between neurons can get stronger or weaker based on activity. The two most studied phenomena are long-term potentiation, or LTP, and long-term depression, LTD.
LTP and LTD. Strengthening and weakening, where are these studied?
LTP, the strengthening part, has been studied most intensively in the hippocampus that's structured deep in the temporal lobe, critical for memory. It's studied both in living organisms, in vivo, and in brain slice preparations kept alive in a dish, in vitro. But LTP and LTD are also found in the neocortex, the cerebellum, and other brain regions.
So what exactly is LTP?
LTP is when a brief high-frequency burst of stimulation like a rapid firing pattern applied to an input pathway causes a long-lasting increase in the strength of the synaptic response in the postsynaptic neurons. This increase can last for hours in a brain slice and potentially days, weeks, or even longer in a living brain.
A lasting boost in connection strength from intense activity.
Exactly. And what's truly revolutionary about understanding LTP is that it gives us a plausible cellular mechanism for how memories might actually be physically encoded and stored in the brain. The thinking is that every time you learn something new or form a memory, the neural pathways involved in processing that information undergo LTP-like changes. The synapses in those specific circuits become more efficient, stronger. It's the brain's way of physically etching that information, making those pathways easier to reactivate later. It's like saying, this connection is important, let's make it stick.
The physical trace of a memory. What happens at the synapse to make it stronger? What's the mechanism?
Great question. The mechanism of LTP, especially in the hippocampus, primarily involves changes happening at the postsynaptic site, the receiving neuron.
Okay.
During that high-frequency stimulation, the presynaptic neuron releases a lot of the excitatory neurotransmitter, glutamate. Glutamate acts on two key types of receptors on the postsynaptic membrane: AMPA receptors and NMDA receptors.
AMPA and NMDA.
Right. Now, under normal low-frequency stimulation, glutamate opens the AMPA receptors, letting sodium ions in and causing a small depolarization. The NMDA receptor is usually blocked by a magnesium ion at this point. But during the intense high-frequency stimulation that induces LTP, the postsynaptic membrane becomes strongly depolarized. This strong depolarization is enough to kick out that magnesium ion, blocking the NMDA receptor channel.
The block is removed.
Exactly. And once unblocked, the NMDA receptor, when glutamate binds to it, allows calcium ions, Kpr++, to flood into the postsynaptic neuron.
Calcium comes in. That sounds important.
Calcium influx is the critical trigger for LTP induction. This influx activates various intracellular second messenger pathways. Key players include enzymes like K+, plus calmodulin-dependent kinase 2, CAM-CANI-IFI, protein kinase G, PKG, and protein kinase C.
Kinases. They usually add phosphate groups to proteins.
Exactly. These kinases phosphorylate target proteins, leading to rapid and potentially long-lasting changes. One major effect is an increase in the responsiveness or number of AMPA receptors in the postsynaptic membrane, basically making the synapse more sensitive to glutamate in the future.
More sensitive input channels.
Right. Additionally, this signaling cascade can activate immediate early genes, leading to the synthesis of new proteins and potentially even structural changes at the synapse, like growth of dendritic spines, which could further stabilize the potentiation for the long term.
So it starts with calcium, triggers kinases, modifies receptors, and can lead to new protein building and structural changes.
That's the core sequence for classic NMDA receptor-dependent LTP. It's also worth mentioning there are other types of synaptic plasticity. For instance, activation of metabotropic glutamate receptors, MGLURs, which are linked to intracellular signaling differently via G proteins, can also lead to LTP or LTD, sometimes by causing calcium release from internal stores within the cell, adding another layer of regulatory complexity.
Okay, so synapses get stronger, LTP, or weaker LTD. That's the microscopic basis. But how does the brain organize all these tiny changes into the memories we experience, from remembering a phone number for a minute to recalling childhood events? Are there different types of memory systems?
Absolutely. Memory isn't a single entity. We classify memories based on duration and content. First, there's working memory.
The mental scratch pad.
Exactly. It's your ability to hold information online, manipulate it, and use it to guide behavior over very short periods, typically just seconds. Think about remembering the start of this sentence while I finish it, or doing mental arithmetic.
Right. How is that encoded?
Working memory seems to rely on persistently active neurons, primarily in the prefrontal cortex. These neurons keep firing as long as that specific piece of information needs to be actively held in mind. We've all had that moment, right? You
Yeah.
walk into a room, then completely forget why you went there.
All the time.
That might be a brief lapse in that persistent activity, your working memory buffer getting cleared.
Okay. Then beyond seconds.
Then we have short-term memory, which lasts maybe minutes to hours, like remembering a phone number you just looked up long enough to dial it, or maybe where you parked your car this morning.
Okay. And then the really long-lasting stuff.
And finally, long-term memory. This can last for hours, days, weeks, or even a lifetime. Long-term memory is often further subdivided into intermediate and truly long-lasting forms. Forming these long-term memories likely involves those more permanent structural changes at synapses, protein synthesis, the consolidation process we talked about potentially happening during sleep.
Consolidation makes short-term into long-term.
That's the idea. And memory loss or amnesia can result either from an actual loss of the stored information itself or from an inability to access or retrieve information that's still stored somewhere.
Interesting distinction. Where in the brain are these different memory types handled? You mentioned the hippocampus.
The temporal lobes, and especially the bilateral hippocampal formation within them, are absolutely critical for forming new recent memories, particularly declarative memories.
Declarative memory, what's that?
Declarative memory, or explicit memory, is your conscious recall of facts, events, places, personal history, things you can consciously declare. I remember eating breakfast this morning, or Paris is the capital of France.
Okay, conscious memories.
Right. The critical role of the hippocampi was tragically highlighted by the famous patient HM, who had bilateral removal of his medial temporal lobes, including the hippocampi, to treat severe epilepsy.
What happened to him?
After the surgery, his epilepsy improved, but he suffered a profound and permanent inability to form new declarative memories. This is called anterograde amnesia. He could remember events and people from before his surgery perfectly well. His existing long-term memories were intact, but he couldn't form new ones.
Couldn't remember anything that happened after the surgery.
Pretty much. He couldn't learn new facts, recognize people he met after the surgery, even if he met them daily, or remember what he did just minutes earlier. Yet his intelligence, personality, and working memory seemed largely okay. It showed the hippocampus isn't where long-term memories are stored, but it's essential for consolidating new declarative memories.
So he couldn't make new conscious memories. Could he learn anything new?
Remarkably, yes. This revealed another major memory distinction. HM and other patients like him could still learn new skills and habits. This falls under the umbrella of non-declarative or procedural memory.
Procedural memory.
Right.
Like skills.
Exactly. Implicit, non-conscious learning. Things like motor skills, riding a bike, perceptual skills, reading mirror-verse text, or habit learning. For instance, if HM was given a task like tracing a star while looking in a mirror, which is tricky at first, he would get better and better at it over consecutive days, showing clear learning. But each day, he would deny ever having seen or done the task before. His performance improved, demonstrating memory, but he had no conscious recollection of the learning episodes.
Wow. So declarative memory, hippocampus dependent, was gone, but procedural memory, hippocampus independent, was intact.
Precisely. It's a fascinating dissociation. The precise brain structures involved in procedural memory aren't fully mapped, but the cerebellum and the basal ganglia are known to play very important roles, especially for motor skills and habit formation.
Cerebellum and basal ganglia for skills and habits. Okay. What about memory for places? Spatial memory.
Ah, another area where the hippocampus and related structures shine. Studies, mainly in rodents but also humans, have revealed specialized neurons critical for spatial navigation and memory. Place cells, found within the hippocampus itself.
Place cells.
Yes, these neurons fire vigorously when an animal enters a specific location, a place field, within a familiar environment. Different place cells have different place fields, and together they form a neural map or representation of that particular space.
A cognitive map in the hippocampus.
Exactly. And it's not just place cells. In the entorhinal cortex, a region that provides major input to the hippocampus, researchers discovered grid cells.
Grid cells.
These are even more amazing. Grid cells fire not just in one specific spot, but in multiple locations that form a remarkably regular, repeating hexagonal grid pattern covering the entire environment. They seem to provide a more abstract, metric coordinate system for space.
Like longitude and latitude lines for the brain.
That's a great analogy. Together, place cells and grid cells, along with other spatially tuned cells like head direction cells, are thought to form the neural basis of our cognitive maps, allowing us to navigate and remember spatial layouts. These maps are context-dependent. They're stable for a given environment, but can generate new appropriate maps if the environment changes or we move to a new place, showing remarkable flexibility.
And this relates to real-world skills.
Absolutely. Think about those studies of experienced London taxi drivers who have to memorize the incredibly complex layout of London streets, the knowledge. Structural MRI studies show that these experienced drivers have a significantly larger posterior hippocampus compared to control subjects.
Their hippocampus grew with experience.
It suggests a structural adaptation related to their intense spatial memory demands. And conversely, difficulty with spatial orientation, like getting lost frequently, is often one of the earliest signs linked to spatial memory loss in neurodegenerative diseases like Alzheimer's, which heavily affects the hippocampus and entorhinal cortex.
It all ties together. Which brings us to this concept of the brain changing. Neuroplasticity. This is truly remarkable, isn't it? The brain's ability to literally rewire itself. How does it happen and when?
It is remarkable. Neuroplasticity refers precisely to this ability of the central nervous system to change its connectivity both structurally, like growing new connections, and functionally, like changing synaptic strength, LTP-LTD, in response to experience or changes in the environment.
And this happens when?
During learning.
Yes. Plasticity is the fundamental basis for learning and memory, but it also occurs significantly in response to brain damage or injury as the brain attempts to reorganize and compensate. And perhaps most dramatically, it occurs during development.
Developmental plasticity. Is that different?
The principles are similar, but developmental plasticity is particularly pronounced during specific critical periods.
Critical periods, like a window of opportunity. Exactly. These are specific, often relatively narrow times early in development when neural connections are most malleable, most sensitive to being shaped by experience. After the critical period closes, the connections become much more stable and harder to change.
Can you give an example?
The visual system provides classic examples. If visual input from one eye is blocked or severely degraded during its critical period early in life, say, by patching the eye or due to a congenital cataract,
Okay.
the connections from that eye to the visual cortex develop abnormally. They fail to compete effectively for synaptic space with the connections from the normally functioning eye. Even if you restore normal visual input later, after the critical period has ended, functional vision from that eye is often permanently impaired.
So you missed the window to wire it up correctly.
Precisely. It reflects this intense competition for synaptic targets during development where connections that are less active or less effectively correlated with other inputs get pruned away, a kind of neural use it or lose it principle operating most strongly during these critical periods.
And this has clinical importance.
Hugely important. It's why early surgery for congenital cataracts is absolutely crucial. If the cataract isn't removed and vision corrected within the critical period, the child may never achieve full normal vision in that eye, even with a perfectly clear lens later on. Similarly, for conditions like amblyopia, lazy eye, or strabismus, crossed eyes, that cause imbalanced input, early detection and intervention like patching the stronger eye or corrective surgery are vital to allow the cortical circuitry for binocular vision to develop normally.
Timing is everything in development. But plasticity isn't only for kids, right? The adult brain can still change.
Absolutely. Plastic changes definitely occur after brain injury in adults, offering hope for functional recovery. For instance, after a stroke damages one area, surrounding areas can sometimes reorganize to take over some of the lost function.
Reorganization. Does it always work?
While sprouting of new axons and dendrites does happen to some extent in the damaged adult CNS, it doesn't always restore normal function effectively. And not all neural pathways seem equally capable of sprouting or reestablishing functional connections. Understanding the mechanisms that promote or inhibit adult neuroplasticity is a massive area of research, vital for improving therapies for CNS diseases, spinal cord injury, stroke, and other neural trauma. There's active research exploring things like growth factors or even using stem cells to try and boost recovery.
Stem cells. Interesting. You mentioned phantom limbs earlier as an example of adult plasticity.
Yes, that's a fascinating and sometimes unsettling example. Patients whose limbs have been amputated often perceive vivid sensations, sometimes even pain, seeming to come from the missing limb. This can sometimes be triggered by touching other parts of their body, like their face or remaining arm stump.
Why would touching your face feel like your missing hand?
Functional imaging studies suggest this happens because the area of the somatosensory cortex that used to receive input from the amputated limb doesn't just go silent. Instead, the representations of neighboring body parts, like the face or the stump, whose cortical areas are adjacent to the hand area, actually expand or spread their connections into that now deprived cortical territory.
So the face map invades the old hand map.
That seems to be what happens.
Yeah.
So when the face is touched, it activates not only its own original cortical territory, but also the remapped former hand territory, leading to the referred sensation in the phantom limb. It's a dramatic example of cortical remapping in adults.
This remap happens in other situations too.
Oh, yes. It demonstrates the brain's continuous adaptability based on sensory input and use. For instance, studies in monkeys showed that after surgical amputation of, say, the middle finger,
Okay.
the cortical somatosensory areas that represented the fingers right next to it, the index and ring fingers, gradually enlarged their representation, essentially taking over the cortical real estate previously occupied by the amputated digits map.
The neighbors move in.
Right. Conversely, in individuals born with syndactyly where fingers are fused together, the initial cortical representation of those fused digits is also somewhat fused or overlapping, but after corrective surgery to separate the digits,
The maps separate too.
exactly, distinct separate representations emerge for the now independent digits, showing the brain refining its maps based on the new distinct sensory input it's receiving.
Use it and shape it.
Precisely. And even more remarkably, this isn't just about injury or surgery. It happens with training and experience. Monkeys train daily for weeks on a task requiring fine sensory discrimination, using just the tips of specific fingers, showed measurable changes in their brains.
What change?
The somatosensory cortical territories representing those highly utilized fingertips actually expanded in size, and the number of receptive fields, that specific skin areas that could activate neurons in that region, also increased within that territory.
So practice literally changed the brain map to devote more resources to that skill.
Exactly. It truly highlights the brain's ongoing dynamic capacity for adaptation and learning throughout life. Your experiences, your actions, what you pay attention to, they are constantly sculpting and refining your own neural maps.
Wow, what an absolutely incredible journey we've taken today.
no.
Just amazing. For the basic, intricate structure of the cerebral cortex, the lobes, the layers, through the secrets of its electrical life with EEGs and evoked potentials, the profound mysteries of sleep, the fascinating insights from how the hemispheres specialize and communicate for language, all the way to this incredible capacity for learning, memory, and that continuous adaptation through neuroplasticity. We've really tried to delve deep into the brain's integrative functions, aiming for that mastery of this complex information.
It's truly mind-boggling when you step back and look at it, and what's perhaps most fascinating is how all these seemingly distinct systems, from those microscopic synaptic changes like LTP we discussed, right up to the macroscopic functions of the different lobes, how they all interconnect and constantly influence each other to create our rich, unified cognitive experience. It's this beautifully complex, highly integrated system, far, far more than just the sum of its individual parts.
So thinking about this, What does it all mean for you, the listener, the learner? Understanding these fundamental principles, getting this deep understanding, it isn't just about knowing cool facts about the brain, is it? It's really about appreciating the profound complexity, the adaptability, and just the sheer potential packed inside your own skull. Every single time you learn something new, every time you recall a memory, every dream you have, these intricate, interconnected systems are tirelessly at work, constantly shaping who you are.
And it really does raise a rather important, maybe even provocative question, doesn't it? Given the brain's absolutely astonishing capacity for plasticity and remapping, which we've just discussed, and seeing the profound impact that experience has on its structure and function, well, what might be the full extent of our potential to consciously shape our own cognitive landscape? How much control can we exert through focused learning, deliberate practice, and maybe even mindful effort?
Yeah. How much can we intentionally sculpt our own brains?
Yeah.
It's a powerful thought. Consider how this knowledge might change your own approach to learning new skills, or perhaps even how we think about recovery after brain injury, or understand neurological conditions. There's always more to discover, and your brain, this incredible adaptive organ, is right at the very heart of it all.